So we are going to go with the topic of inference and comparison of biological networks. We kind of touched on this in the previous session, but I think you're going to get complementary discussion this time. And Kimberly is the one who's coordinated that along with her team.
And I will do my best to keep track of the time, so we have plenty of time for discussion. There's the laser pointer. All right, thank you. This was supposed to be session one, topic one, but we're doing it now. It's inference and comparison of biological networks. And when we were meeting as a group, we kind of settled on four main subtopics within this. And so we're going to break this time up into these four main subtopics. And they're roughly ordered and something that we thought was logical. Of course, they all kind of feed into each other. But we're going to start with the inference of networks, because the idea is before you can do anything, you've got to have a network from somewhere. So how do people actually infer networks? Next, we're going to go into link predictions and their applications, which is highly related, right? What if you have a partial network and you need to try to build in some missingness? Finally, next we're going to talk about condition-specific subnetwork inference with the idea that if you already have a network, then you have some data, how do you figure out some sort of important subnetwork within that? And then at the very end, we're going to talk about differential network analysis. Let's say you already have some network and you want to compare them. How is that done? How should it be done? Where is the field going? So subtopic one, inference and networks. These were the slides that I had put together. And I will say this is kind of just a high-level overview of the field, kind of what people often talk about in this area in my mind. All right, so how do we infer networks from data? And in particular, I'm mostly talking about omics data in this context. There's, of course, lots of other ways to infer networks. We heard some about biomedical knowledge. But in this case, you can think about having a biological network. You can't really directly measure it. But what you can measure is you can measure omics data, right? You can interrogate the cell. And what we want to do is we want to use that output of the network, the omics data we can measure, to kind of reverse engineer or infer the underlying network. This is an overused cartoon, but I actually do think it's very apt for this. What essentially happens is different people have their favorite omic, or different populations have different omics measured on them. And basically, we're looking at different parts of the elephant, and we want to reconstruct the elephant, which is the underlying network, with incomplete data that's only an outside observable. We're kind of like these blind men. All right, so building and interpreting biological networks. I wish I could see these words better because I want to actually talk through these. I think these are some important points. On the right are different types of biological relationships, the types of things we might want to reconstruct, or the types of things we would want to infer. And what I said earlier, biological networks often aren't known a priori, but you have to infer them using genomic data. How a network is inferred or reconstructed, you'll hear me use those interchangeably, often depends both on what data is available. If you only have, transcriptomics data or only epigenetics, that's going to dictate how you build the network. And what you want the edges in that network to actually represent. Do you want them to represent causal links or correlative links? Okay. Are they actually going to be physical interactions or some sort of other type of interactions? And finally, the meaning of edges in a reconstructed network depends on the method that is used to generate those edges. That's important for downstream analysis and interpretation. If you're doing something like a correlation network, you don't know causation. It determines how you can actually interpret those. All right, interpreting the edges in biological networks. This is kind of a very high-level, crude thing, but I kind of like to break down networks into three main types. The first is relational networks, which tend to be undirected. A good example of this are something like protein-protein interactions. Now, of course, those can theoretically be directed, but in the high-level sense, you think about two proteins that come together, they dimerize. It's an undirected physical relationship. The second is what I call a correlation network. That is also undirected. Two things go up and down together, but you can have signs in this, right? They could be negatively or positively correlated. And then finally, the third type that I like to kind of group things into is what I call a regulatory network, where you have things that are directed. A causes B to do something. This can have signs, although these are typically hard to infer in practice, but something could say upregulate or downregulate a target gene. So I'm going to start with correlation networks. There's lots of caveats and limitations to correlation networks, but I think it's an important topic to kind of discuss. So how do correlation networks normally work in the very high-level sense? Well, the basic approach is that you have something like gene expression data. It doesn't have to be gene expression. It can be metabolite expression, CpG methylation, any omics data. And you basically use a similarity score to reconstruct a matrix. Similarity score could be Pearson, Spearman, mutual information, you name it, your similarity score, your choice. You threshold it, and you analyze it. I put over here 3 old algorithms from this field, and I don't want to go into them all in depth. They're pretty old, but I put them up here because they're still constantly referenced in the literature. WGCNA is weighted gene co-expression network analysis. People use this a lot. Basically take Pearson correlation networks, scale it, and find modules of genes. Arachne used mutual information, and the idea was correlation's not causation, so let's kind of snip edges to try to get rid of indirect interactions. And the CLR did an extra normalization procedure. And there have been a lot more since then, but again, these are the straw men that everyone likes to cite and then knock down for their new method of choice. So I wanted to go over them briefly. All right, correlation networks are very, very widely used in biology. We can all nitpick correlations and not causation all we want, but they're ubiquitous, right? And these are just 4 examples I grabbed from the literature to show how widely they're used. Up here, these are nodes are genes. Down here, there's an example of correlation network where the nodes are metabolites. This is a paper I was actually on where the nodes are foods, where people were trying to study nutrition and diet. So they said, what do we do with this? Let's build a correlation network. And down here, these are nodes that are biomarkers, where you have a bunch of patients and nodes are measured and people looked at correlation patterns. So they're very, very widely used, including in recent publications in decent high-end journals. All right, but as I keep on iterating, and we all know correlation is not causation. So let's take this example here. You have 2 genes that are correlated. We know that co-expression can occur when, say, one gene regulates another, right? Could go in either direction. We don't really know. There's some sort of connection. The other reason it can happen is you have something like a regulatory protein that targets 2 genes, and voila, those 2 genes are now co-expressed, right? So both represent the same way in a co-expression network. You can't really disentangle these very easily. Which leads me to my second type of network that I'm going to touch on in this section, which is regulatory networks. And in particular, I'm going to talk about gene regulatory networks because that's the one I know the most about, but this could also be things like metabolic regulatory networks and things like that. All right, regulatory networks. All right, so gene regulation is incredibly complex, and if we weren't trying to cover every single topic in this field, I could probably spend 10 minutes on this slide talking about all this different data and all the different caveats and what it means. But at the end of the day, what actually happens is we have different types of regulatory mechanisms, protein interactions, chromatin structure, chromatin state, methylation, microRNAs, protein expression, and we want to reduce that down to something that we can model as a network. And when I talk about gene regulatory networks, I normally talk about transcription factors regulating target genes. And some of the target genes are also themselves transcription factors. All right, so how have these been modeled? I like to think about there being 2 main ways to actually produce this type of network in a reasonable way. The first is experimentally. If you had infinite amount of money and infinite amount of resources, you can imagine taking transcription factor antibodies, interrogating the entire genome in your particular cell of choice, and building this network. And that would work, but it has a lot of limitations. First, it's very expensive. None of us have infinite resources. And also, there's actually a limited number of experimental antibodies you can use to actually build these transcription factor gene connections. But the strength is it's high quality and it's context specific. The second is computationally. You can take something like a transcription factor and you know something about where it might bind in the genome based on its sequence. And say, okay, we know that there's a DNA sequence map to the transcription factor here, the promoter region of a gene. Let's just say that transcription factor regulates that gene. There are still limitations. We still only know these recognition sequences for about 50% of transcription factors, and they're of various quality. And also, it's very prone to false positives and not context specific, but it's really cheap. You have a high-end computer, you can run this, in a matter of hours these days. So what's actually happened for gene regulatory networks is we've had a combination. We want to take something like experimental data and combine it with computational predictions. So I'm going to touch on 2 of those, and then we'll go on to our second subtopic. First, methods that use gene expression data. And this is a figure from an old paper now from 2012, where this was actually one of the dream challenges. So someone from SAGE mentioned this earlier. This was an example of that, where they actually had data they provided to the user base, where they said, put your algorithm of choice, try to predict this gene regulatory network, and then they benchmarked it systematically. So on the x-axis are all different methods, the y-axis is the AUPR. And silico data, they did pretty well. E. coli, they did pretty well, but when they got down to yeast, in my opinion, that looks like nothing really worked. And I showed this slide to people again and again and again, because even though it's been 10 years, I think that for the most part, people who try to apply algorithms to high-level organisms will see this trend, that they're really, really, really hard to predict. But that being said, it's worthwhile to mention that there are a number of methods in this area. I'm mentioning 3 here. I'm mostly mentioning these 3 because they're highly cited, and I see them benchmarked against again and again and again in the literature. But this is an explosive field. There are a lot more out there. So inferolator is one that is still well maintained, that uses linear regression, ON shrinkage, and lasso to basically combine transcription factor to gene predictions with gene expression data. Genie3 that uses tree-based and ensemble methods. And then Panda is one that my group developed that actually tried to integrate multiple types of omics data to find consistent structures that way. The second type of omics data that I've seen a lot in gene regulatory networks is epigenetic data. So in this case, we have an example where I talk about here there's a DNA, and you have predicted transcription factor binding sites along it and open chromatin. You can imagine you can use that to basically prune the sites and thus prune your network. And this is an approach that is taken when you have high-quality epigenetic data. And then going forward, here are just a couple papers and resources that use that approach. This was one of the earliest papers that came out that did this. They had DNA hypersensitivity and a number of cell lines, and they tried to build transcription factor to transcription factor networks in each of the cell lines. This is a more recent approach, TEPIC, that basically combines transcription factor binding affinities with annotation data, including distance from the TSS and chromatin data. And then SPIDER basically identifies consistent structures using the Panda method, but taking into account the epigenetic context. And so those are the gene regulatory networks and the co-expression networks. I'll end with some discussion points, and then we'll move on to the protein interaction networks and link prediction. So first off, correlation and co-expression networks are easy to use, and they're widely applied, right? We can't avoid that fact. They're very easy and used a lot. However, they have multiple limitations. Correlation is not causation, and it's difficult to really know what the edges mean. On the other hand, gene regulatory networks are really difficult to infer. And also, there are a ton of methods, but the accuracy remains low. And it's very challenging to know how to take all that omics data I talked about and actually use it to infer a regulatory network. All right, I believe the next subtopic in the same theme is Mona and Lenore, and they will be.
Lenore will be able to present. Just tell us when.
Okay.
Yeah.
I'll try to coordinate. So Lenore and I decided to tag team this, which would work perfectly if we were here, but maybe a little more complicated.
Can you use the mic so that Lenore
Okay. And how do I? Which one is that? Is this the? Oops. Oh, I see. How do you go backwards? Got it. Okay. I'm going to go to the next slide, and Lenore is going to start off.
Great. Okay. So Kimberly has given a beautiful introduction into regulatory networks. I am going to switch gears and go back to sort of the classical physical protein-protein interaction networks. And these are the networks that are built from trying to, from experimental evidence that protein chains physically bind in the cell. Here we have an example of a protein complex, and we're going to abstract out all that wonderful 3D structure information and just basically say this little purple thing binds to the orange thing, which is in a clique with the blue thing and the green thing. And if you'll notice, this is what you would get from like a yeast to hybrid type experiment. If I was doing a co-IP experiment, then actually it witnesses co-complex membership. So I would actually also get edges between the purple and the blue and the purple and the green. If I look at all the different experimental interactions in one source, BioGrid, you can see about how many of them have been collected in these public databases. And you also see that humans are where we have the most, though I guess if you mod out by the size of the genome, we also have significant in yeast, and everything else we have much fewer. And once we get beyond just a few model organisms, we don't have any data. Next slide. Okay, so as many people in different sessions today have already alluded to, in these types of networks, there are many different ways that people use the existing graph theoretic interconnection structure of the network to predict possible additional edges. So when we get this biogrid picture, we already assume some of the edges are wrong because they're experimental error, but even more, we assume that many, many edges are missing because not all pairs of proteins have been tested for interaction. So therefore, inferring additional protein-protein interactions computationally can be extremely useful. Very simple methods include just counting the number of common neighbors. Other simple local neighborhood methods like Adame Kadar, other things have been used also in social networks. Going to say something a little bit later about this L3 measure that was introduced by Kovacs et al. And a lot of people have already been talking about random walk diffusion methods, network propagation methods, different ways of taking short random walks in order to see which nodes are kind of hit most often and in the neighborhood and nearby. And there's subtleties about the different ones of these methods that people use in different settings. Embedding methods often also have underlying a kernel that is random walk based for these methods. Glide is a method of ours. There's now glider as well. And this little schema is from the review paper that I did with other people who are at this workshop, either here or at Zoom, where we looked at all the different ways that sort of random walk-based methods have been used to date in biological networks. But since then, they've gotten with, so Ben Raphael was involved in that work, as was Roded Sharon, who actually Roded convened us, and they're also participating in this workshop. So next slide. So this is just sort of highlighting the fact that which metric you use to talk about which nodes are most likely to have links can actually affect very strongly what you do. And it can be different than what you might want if you're asking a different question, such as functional relatedness. So the common neighbors measure. So here's a gene PARC2, and we're looking at its closest neighbors, 20 neighbors, using sort of the common neighbors measure. It's a weighted measure that I am completely describing, but basically you're closer by this measure if you have many common neighbors in the network. And by this measure, these are the proteins that are closest to this gene PARC2. Actually, this is the ones, I'm sorry, they're closest to the gene PINK1. PARC2 is also a very well-known Parkinson gene. And we're looking at the closest genes by this weighted common neighbors measure. And you'll see that in terms of the known links, these genes are quite disconnected. There are even some disconnected genes. And the reason is, of course, there's some other nodes in there that they're mutually connected to, but there's a lot here that isn't directly connected to PINK1. On the other hand, this recent measure L3 of Kovacs et al. Uses sort of in a graphlet sense a very different interconnection pattern, which pays attention to the fact that protein-protein physical interactions more often have kind of a lock and key mechanism, where you would assume the locks bind to keys, but the keys would not bind to keys, or the locks would not bind to locks. So by this L3 measure, when we look at the closest 20 genes to this pink one, we get all these genes that in fact are known to be direct interactors with pink one. And this gene over here, which maybe now I predict is a direct interactor with pink one. So for actual direct link prediction, it might actually matter a little bit more which version of random walk, which embedding, or which measure of similarity I use. Okay, I'm going to turn this over to Mona.
All right, my turn. So I'm going to, follow on Lenore's nice introduction. So I wanted to start by saying that, the methods that Lenore's been talking about for link prediction are largely based on exploiting or leveraging topology in these large-scale protein interaction networks. And it's really amazing that just the topology of a network actually reflects so many important things about biology. So for example, it was shown, from the very early days that high-degree nodes tend to be essential. That's been something that's been seen over and over again. Or, and we've already heard about this, I think, earlier in the morning, we know that if you look at these protein interaction networks, there's a structure to them. They have modularity, and then these modules within these networks, which are just, again, defined topologically, correspond to coherent biological processes or complexes. These topological features can also actually reflect dynamics of biological networks as well, or of proteins. So, for example, if you look at the clustering coefficient of a protein, if that number is high, then in general, the protein is going to be co-expressed with its neighbors in taking part of a complex. So it can actually represent, topology can also represent dynamics of proteins. And then people have shown that you can identify various aspects of disease genes, or you can use as a feature to identify disease genes. And we had an example this morning as well by just looking at differences in topology of disease genes. But I think if you, so then it's natural to think about link prediction with topology in mind. How can you use topology to close out and find these links that we're missing, or maybe even to get rid of erroneous edges that are there? But I think it's important to look under the hood a little bit and see that these nodes have various types of well-known features. And this is, before getting too deep into sequences, which we'll talk a little bit more about in a second, I think. People have determined, for example, particular annotations over the years that are associated with these proteins. They've also know things about structures. They've also, labeled these proteins with respect to what domains or peptides of functional importance that they contain. So one thing we've thought a lot about in the past is when you think about these networks, can we extend the topology-based measures just a little bit to include some of this additional information? So there are also these recurring patterns, we heard about network motifs in the morning, that are purely topological-based units. But you can also have an extension of that where you have a topology and you have an annotation on a node. And this is something that we've called network schemas in the past, but they're a natural extension of network topologies. And so on the left here is a schema. It's just a straight line corresponding to something that's related to RAS signaling, where this RAS protein is regulated by this GAP protein, and it in turn regulates this kinase. If you look at a protein-protein interaction network, and we're talking about physical interactions here, you would see many such instances of this schema. And so this is a little piece of a yeast interaction network from a while back. But you can look at higher order combinations as well. And so you can look at pairs to start, triplets as we just talked about. You could look at triangles, which maybe help capture complexes. And you can look for kind of branching decisions starting as these Y stars is what we call them. And so you can ask, if you look at a large-scale biological network, do any of these network schemas tend to be enriched within them? So at the pairwise level, it's clear there are many of these common patterns that you'll see when you look at any biological network. In this particular example, it's looking at the yeast protein interaction network, and it's just looking to see pairs of PFAM domains that are enriched in these networks. And of course, many people have looked at domain-domain interactions in the past. And so this is another type of network. This is the network of annotations that tend to recur more often than you expect in the protein-protein interaction network. But you can do this. Oh, and if you look at them, these things make sense. So here are some pairwise schemas that are involved in signaling. And here's a sub-part of this network that's involved in transport. But you can look at higher order topologies as well. And here what you would see is that there are higher order schemas that also repeat, and they repeat, and they're statistically enriched even when you correct for the pairwise schemas that you observe. So, we have multiple of these schemas that recur in interaction networks, and multiple of these schemas that recur more than you would expect based on their pairwise compositions. And this is just looking at PFAM domains, and of course you could do that on any annotation type. And so I think a very interesting thing to think about for link prediction, at least for topological-based link prediction, is to think about link prediction with respect to topology, but also to these very simple annotations that exist. And I think that those would be ways in which link prediction methods would certainly get kind of more power. And so I want to make a little bit of a distinction here between link prediction when you're looking at protein-protein physical interactions versus link prediction when you're looking at the types of co-expression networks that we just heard about, or functional networks. So you would tend to see these types of patterns that correspond to, say, a signaling cascade in a protein-protein physical interaction network, but you wouldn't expect to see the same types of patterns in a co-expression or functional network. So depending on the type of link prediction you're trying to do, depending on the type of domain that you have, these types of annotations would be more or less important. But to me, they're a really natural way in which to start thinking about extending link prediction when you're using topological measures. And I think we're back to Lenore. Yeah.
Okay. Well, and now for something completely different. Rather than looking overall at the network, if we're predicting protein-protein physical interactions, we have a lot of individual data in the protein chains. In fact, we have the protein sequence, and from the protein sequence, we have the protein structure. And in fact, it is possible to take a pair of protein sequences and devise a method that can predict just based on the pairwise protein sequence data if there is a predicted interaction. And a lot of the methods that people have been using to do this are based on protein language models and deep learning using some kind of neural net architecture. And there are several methods out there, including Piper and others, which we will discuss in a bit. Next slide. So our offering in this space, and this is joint with Bonnie Berger's lab, is we've worked on a method that we call Descript, which seems to be better at predicting PPIs in a new species when trained on a different species than some of the other deep learning methods, which do very well when trained on human data and then are asked to predict PPIs in human data, but other methods do less well when trained on human data and say asked to predict PPI in fly or mouse. So this is just a little schema of the architecture of the model. We try to be very interpretable. So it's doing two things. One is that it's leveraging already some kind of structural information that comes, as Mona said, from PFAM, and some knowledge of structural domains that are embedded and learned by the language model for individual sequences. But then it tries to somehow learn a contact map between these sequences. So, and that makes what it's doing a little bit more interpretable and a little more generalizable, because it probably has to learn something from structure if it's trying to learn sort of how they interact. So we never read that out. the readout of the model when it's end-to-end trained is just the binary interaction prediction. And but this is some sort of evidence that in the middle, when it's saying, yes, I think these two proteins interact, that it really does somehow have some sense of what that interaction surface looks like. Okay, next slide. So at ISMB in July, we're presenting new work that tries to combine these two views. So one question is, if I could do a network prediction and I could do a sequence prediction, how could I somehow leverage them both? So the first way we ask this is in a setting where we don't have PPI networks at all. So maybe we're trying to predict PPIs in a non-model organism where we have no existing experimental evidence. But we're still somehow guiding the training by this global knowledge of network topology by one of the embedding methods. So somehow, when we're learning what makes two proteins interact, somehow we're learning something from the structure at the bottom level, and somehow we're learning something about maybe which nodes are most promiscuous or various other things from the network level. But again, this is topsy-turvy is a method that doesn't need any network information to predict. It just takes as input two sequences and predicts. It's just in training that it's somehow learning something about the network. Next slide. And then if you're in a species where you have both, where you have like humans or fly or any one of those species I showed in the beginning, you might want to ask, well, is it better to predict interactions, missing links from the network, or is it better to predict them from sequence? And of course, as you would expect, somehow we want to combine, right? We want to predict from sequence when we're not confident in the network, and we want to predict from the network. So some people say the sequence really tells you in a social network analogy which two people are compatible to be friends, but the network tells you which two people have actually met. So you do get different kinds of information from this kind of data. And in fact, we're able to do even better at correctly predicting links and cross-validation using this TT hybrid new method that uses both the topsy-turvy on just the sequences and then our glider method on the networks. So I do want to make, so this is early days. I mean, this is a method that we don't know a lot of methods that do both, but I think this is a great open problem to refine and improve these kinds of methods. I would caution that the issue of ascertainment bias becomes a little bit dangerous when trying to evaluate results. And it's really hard to measure or compete a method that's working with sequence data against a method that's working with network data on the current network data sets we have. And the reason is that, in fact, the experiments that the biologists have done are biased by pairs of protein they already think maybe interact, or proteins in the neighborhoods of some very important disease proteins or other proteins like this. So as a result, we've probably overrepresented the popular edges that network methods would infer, and we've probably underestimated true PPI sequence-based pairs that we haven't tested. Now, there's HURI, which is the Human interaction network that of Luck et al. That recently came out that does a completely unbiased yeast to hybrid screen of a portion of human proteins. So if you start with purely unbiased data, then you might not have this problem. Or you could look at other ways to sort of ameliorate the ascertainment bias if you want to compare these two types of data. Okay, I will stop there and back to Mona.
All right, so I wanted to switch just for a second from networks to sequence. So, we know that there's a lot of variation across the human population. We look around the room, we can see a diversity of phenotypes here. And these, of course, depend on genomic sequence. And so if you look at our genomic sequences, there are places in the genome where we vary. We're mostly the same, but we also vary as well. And then, of course, this is also super interesting in the context of disease, whether it's germline variants in inherited diseases or it's acquired mutations in diseases such as cancer. And one important thing I think we want to think about as a field, we've talked a lot about like context-specific networks and temporally changing networks, but there's also networks across populations, whether healthy populations or disease populations. And so I think it's really kind of important to think about the effects of variation and mutations on networks themselves. And so just taking a protein-centric view here, you can think, to start anyway, you can think about a protein sequence, and you can think about a mutation, and maybe that mutation actually adds an edge to a network. Maybe you have another mutation, maybe it changes the interaction between those two proteins, I think P3 and P4. You get another mutation, and maybe that's neutral, it has no effect on the network at all. Another mutation, and maybe it's so detrimental it kills the entire protein, affecting a whole bunch of interactions that you have. You can also have mutations outside of protein sequences, of course, and so you get a mutation in a regulatory region, and maybe that removes this regulatory interaction between the regulator and its target gene. So just thinking about this variation, you have to look a little bit as to see where the variation is and what the specific alterations are. So certain positions or mutations are important for interaction networks, and this comes partly to trying to understand the 3D structure of proteins and how they interact with ligands. So of course you can imagine that the positions that are in contact with the ligand are going to be more important for the interactions that they make. And so I think one of the really exciting things to think about in the future is to try to predict these personalized networks. And there are kind of two ways to think about it. On the one hand, you can do predictions sort of de novo. So the types of things that Lenore just talked about, why can't we go and look at all the variants and see how they would alter various kinds of interactions, whether it's protein-protein physical interactions or protein DNA interactions, protein small molecule interactions, et cetera. The second type of approach is, well, we already have a lot of information about networks already. How can we predict the impact of variation or mutations on existing networks? And so that's an approach that we've taken both in the disease context and shown that these disease proteins tend to accumulate mutations in positions that participate in interactions. But we've also done it in another setting, and so I just wanted to tell you a little bit about that before stopping. And so here, we thought about, and this is work we just published in Recom, about predicting the impact of mutations on protein DNA interactions. And here we started off, if you look at the left, how does the pointer work? Which one is it? Okay, there you go. So, from studies like ENCODE or other large-scale studies, we have lots of examples where we know a protein and we know kind of the binding preferences of this transcription factor protein. But what we don't actually know is what this interface is like. So we have the edge, we don't have the interface. In many cases, we can have models of what this interface is like. So here's the protein side. These are different positions. And it's not that all of these types of protein-DNA interactions happen this way. This is for this homeodomain class of proteins, but a lot of them do. So you can come up with contact maps for certain restricted cases. And so then we showed that you can, if you have a contact model like this, and you have kind of data that looks like this, you can actually learn energy terms and this kind of registration on a contact map simultaneously. So you're learning a structural interface. And then in the end, what that means is that once you've figured out the structural interface, and this is structural interface loosely defined, meaning I know how this protein, what its rough contact map is with this DNA motif, then you can think about whether you have a mutation that occurs in a particular protein position and what its effect would be, or which positions of DNA it's likely to affect in terms of binding. So this is just one type of way that you can think about mutations and the effect that they have on known binding interfaces. So overall, I guess I'd like to say that I think that the field as a whole should start thinking about the impact of variation on networks, and particularly thinking about like the diversity of populations that we have. So we have many different alleles, and there are whole families of proteins that are hyperpolymorphic, like MHC, and trying to think about methods for predicting these types of interactions when we know maybe roughly what the interactions are like or which things interact with each other, but what the impact of this variation is. And so I think that's it for me, and I'm passing it on to Ben.
Okay. The lasers on the back. Perfect. Okay, great. So we've, at least in our group thus far, talked about constructing networks, talked about predicting new links in networks. And so I was sort of tasked to talk about what we called condition-specific network inference, which is really how we use networks to really interpret and analyze other types of data. And we heard a little bit about this morning, and this sort of problem arises in many applications in analyzing biological data. I just put a few here. Sort of the classic function prediction problem, which is, all of these are premised on starting with a biological network, say a protein-protein interaction network. And so if you have such a PPI network and you know something about some genes here being implicated in I can't even read what that is, metabolism, this ankle, then you might be able to say that other genes that are unannotated in the same subnetwork will probably also have that same function. Or if you've perhaps done a large-scale measurement, like maybe you've done a genome-wide association study, and you identify that a few genes here in red are sort of have a significant association with height in this case, but then by also using an interaction network, you might be able to pull out some coherent subnetwork that involves many other genes which were not significant, didn't rise to the level of statistical significance, and this might be sort of some indication that it's this group of genes, this subnetwork that is associated with height. And a similar story in cancer, where you might look at somatic mutations, and many of these mutations are rare, and so you might not be able to identify all of the cancer-causing mutations just by their frequency or statistical significance individually, but if you combine them on top of a network, you might be able to then pull out sort of coherent subnetworks of cancer genes. So, this is a problem that's been well worked on by members of the community in this room, many others, and I think we've maybe already seen both these review articles listed, and there's actually even dozens of review articles with many methods on this topic. And just to sort of describe them at a high level, I would sort of classify them in really just sort of two major approaches, which I'll refer to here as network ranking and altered subnetworks. And so in both cases, you start with some interaction network and some labels on the genes. On the top here, these labels are binary. They might know the function of some genes and not know the function of others. And then in this network ranking approach, I then use some measure of network similarity. We've heard about a few of these, and I'll talk about these in a second. And then some sort of algorithm that runs through the similarity measure, and then we'll rank the genes according to their similarity. And I can sort of threshold this and maybe get some prediction about which other genes have that same function, for example. Similarly, but slightly different, I might think of having a score on each node of the subnetwork. It could, again, be binary, or in this case, it's shown as some sort of continuous score. Again, through sort of network similarity, I pull out, in this case, sort of these high-scoring subnetworks. Okay, now, I sort of showed this in a slightly different way because, of course, all of these nodes are actually on the network. So I could really think of having these rankings on the subnetworks as well, except there's sort of a subtle difference between these two approaches that I'll discuss in a slide or two. Now, these similarity measures we've heard already a lot about, and sort of historically, maybe the simplest was to just look at direct neighbors and then sort of shortest paths. We heard about this morning. And then sort of more sophisticated measures that sort of propagate information on networks using random walks or diffusion or sort of network flow approaches. And so, again, we've already seen this and heard about this. So network propagation has really become one of the most popular approaches, I would say. And that really, in and of itself, is a term that actually encompasses numerous such approaches that involve sort of random walks on graphs, as shown in this approach. Equivalently, many of these random walks can be described as fluid or sort of heat diffusion on a graph. And we showed, Lenore already mentioned this review article, sort of how really a lot of these approaches, what they really boil down to is this process, which is often described in this iterative way of, you take iterative steps of a random walk or flows, or really just can be written down in terms of a network similarity. So the random walk, with restart, diffusion kernel, nearly everything people write down really just is a similarity matrix. And network propagation, this becomes as simple as matrix vector multiplication. So it also makes it highly efficient. I just wanted to highlight kind of one sort of twist on this, which is sort of using kind of this propagation approach twice to really merge both those ideas. So one where you might have these binary scores of prior knowledge. This is actually work of Mona's, where you can sort of propagate this prior knowledge and then use that in and of itself to then define a new random walk, which then you can take your new information, say your somatic mutations in cancer, sort of random walk again and get sort of another prioritization. So there are many, many variations of these ideas. However, and this is the point I wanted to mention about the difference between ranking and subnetwork identification, is that often, especially with the more sophisticated measures, what you are really getting is just the ranking. Okay, so network propagation actually just gives you a score on each vertex. In and of itself, it doesn't pull out a subnetwork. So you actually need some other sort of scheme to decide that maybe this is your high-scoring subnetwork following propagation. And if you go dig into these papers, and I pulled out sort of two, one from Brodich Sharon's group that sort of describes how then after propagation, they go pull in densely connected subgraphs. Or in our approach, we said go back in and pull out these strongly connected components. So there's some other heuristic going on to get the subnetworks. It's not really just that flow measure. And this is an important distinction because it really says that the topological properties are being used in a way twice and sometimes differently. So it's the random walk that's being used to change the scores, but then maybe it's actually cliques that are being pulled out of the network after these scores. Okay. So there's a lot of sort of subtleties under the hood. And so, because of some of these subtleties, we sort of wanted to see if there was a simple generative model we could write down to sort of encompass what a lot of these approaches were trying to do. And so what we came up with is what we call the altered subnetwork distribution. So a very simple statistical model that's very flexible. So you start with your interaction network, here a graph. You have this subnetwork family, which I'm going to talk about in a second. This is sort of your prior on what you think the interesting possible subnetworks could be. And what you want to find is an altered subnetwork from that family. Okay? You're also then given these vertex scores. And the vertex scores are distributed in two ways. They have a background distribution here, or for nodes that are in this altered subnetwork that you're going to implant, they have this altered distribution. Okay? So now you've got sort of these parameters you can use to describe different approaches. What is the subnetwork family? And what are these background and altered distributions? So for simplicity, you could think of these scores as coming from two normal distributions. And this becomes like a network mixture model, if you will, where the background model is a normal distribution with mean 0, and then in your altered subnetwork, your scores are higher, so you have some higher mean mu. Okay? The subnetwork families, you can then think of all the different approaches people have tried. You could think of connected subgraphs, pulling out connected subgraphs with high scores, or maybe you want edge-dense subnetworks, so you want cliques, or maybe you want graphs with minimum cut size. Maybe you want somehow propagation subnetworks, whatever that means. But now, by writing things down this way, we have a version of the problem that's really a statistical estimation problem, because then we could say, all right, given a graph, and given the subnetwork family, now from vertex scores, find the altered subnetwork. Okay? So now all that is a problem of estimating the parameters of this altered subnetwork distribution. And then by changing the subnetwork family, or by changing these distributions, I can then model various different problems that have been presented in the literature. And then what we can do is start to describe which versions of this problem are easy and which versions are hard. So thinking first about the vertex scores, if the background distribution and the altered distribution for my subnetwork are pretty far apart, then that's a really easy version of this problem. In fact, so easy that I don't even need the network anymore. I don't need the network information. It'd be very clear if I want to separate two normals. If their means are very far apart, I don't need this prior knowledge of the network. So that's easy. Of course, if those distributions are completely overlapping, the problem is hard, or maybe even impossible if my subnetwork scores are not much higher than my background scores. And then on the other axis, we can think about the subnetwork family itself also. So if the subnetwork family is really large, in the limit, we could think of the subnetwork family including all sets of nodes, 2 to the n, right? And then there's no structure at all. So of course, that's a really hard version of the problem. Or we can think about the subnetwork family involving one subnetwork. And that's a very easy version of the problem. In fact, that's just equivalent to doing like a test between two sets of nodes, like a t-test if it's the normal distribution. So now you have these two axes to think about easy and hard versions of the problem. And so the sort of interesting versions of the problem are somehow right here in the sweet spot in the middle. Okay? So you can think about where your problem is it in the sweet spot, compared to very easy versions of this problem, which are what we call the scores-only version, where the distributions are so far apart that you don't even need the network, or the version where the network alone is enough and you don't need the scores. There's only one subnetwork. You could just test them directly. Okay? So these create interesting baselines of comparison. Right? There's a lot to say. I'm just going to actually give 3 sort of summary discussion points here in the interest of time. The first one is that actually many studies, we claim nearly every study, doesn't compare to the correct scores-only and the correct network-only baseline. And by correct network-only baseline, what I mean is that you have to use your network information in the same way that your algorithm is using it. Okay, so for example, if I do network propagation with random walk with restart, then I should compare to page rank centrality, which is random walk with restart without any scores, to show that my network method is doing better than baseline. If I compare to like direct nearest neighbor interactions, then I haven't sort of said whether my algorithm is doing better because it used a different network similarity, or if it's doing better because it actually did something interesting for this problem. Okay, so just to show you, we sort of reanalyzed some data from a study that had done network propagation with GWAS or the network, and we showed that actually when you looked at in this way, for what did they do? 8 GWAS studies, 3 of them you could just get the same or better performance only using the network, meaning you didn't even need to do the GWAS to pick out the genes for that study. They were already encoded in the structure of the network. Or the GWAS alone was enough, and you'd actually do better by just ignoring the network and just looking at the GWAS p-values and ranking genes. And it was only 3 of the 8 where actually both pieces of information were useful. And this is all sort of with a specific network propagation, random walk with restart type of centrality. But now you can do this with, choose your favorite algorithm as this last part and see if you can do better than these 2 baselines. That's one point. The second point is that many of the algorithms that we've developed, and I say we meaning even my group, so not us collectively, don't really have good statistical guarantees of when they work and when they don't. And so we investigated this in using this sort of altered subnetwork distribution type of framework for one of the seminal methods from Trey Addiker's group called J-active modules, which was a method that was developed for gene expression. And what it did was it found maximum scoring connected subgraphs. Okay, so now we know in our terminology, that means the subnetwork family are connected subgraphs. In fact, the scores were normal scores because they use z-scores. What we actually could show is that this score that they used was in fact the maximum likelihood estimator of the statistical problem, which sounds good. They're doing maximum likelihood estimation, which they didn't even know in the original papers. It's kind of cool that that's what they did. However, that maximum likelihood estimator is biased. And you can actually show this. What that means is it explains a phenomenon that had been known for 15 years or 20 years since this algorithm existed, which is that it tended to produce very large subnetworks. And it's actually just a feature of the statistical problem and the fact that they're using this connected subnetwork distribution. And then the third summary point that I'll sort of say is that the choice of this family, now that you expose all the parameters of the algorithm, then you can sort of ask, well, what topological assumption should we use? That corresponds to, in this statistical estimation problem, which subnetwork family should we use? Do you want connected subgraphs? Do you want dense subgraphs, minimum cut? Do you want sort of somehow propagation? What does it even mean to have a subnetwork family defined by a random walk? And thus far, I would say, a lot of our algorithms are really make this choice just empirically, or just almost even randomly, what seems like reasonable. And I think we should maybe think about which of these choices make sense in different particular applications. So I'll stand there and we can have some discussion maybe on some of these points. I think I did my 10 minutes, so I did my job.
I think you did. All right, I'm going to wrap up with subtopic 4. I'm going to try to keep it to our agreed upon 10 minutes if at all possible. And Holly and I are tag teaming this. I have the first set, and I'm going to turn it over to her to wrap it up. So our final subtopic is differential network analysis. And I was going to start with a quick story. I'll make it brief. I like to introduce it this way. Let's pretend you have two patients, Susan and Jane. They grow up, they're healthy, they have various habits throughout their life, such as eating burgers or wine. As they get old, they have a similar disease, similar phenotype. They both go to the clinic. The doctor gives them the same drug. And lo and behold, you can see if you look closely, Susan gets better. She's smiling again, but Jane doesn't. So the underlying hypothesis of differential network analysis and why we compare biological networks is this. We can say that early on, both Susan and Jane, even though they have different networks, they're both healthy, they're both functioning. But as they get older, their network develops mutations, perturbations in such a way that it becomes diseased. But they become diseased in different ways that even though they have a similar phenotype, they might respond differently to something like a therapy. So differential network analysis, kind of at a high level, is generally done in two ways, at least in kind of the omics inferred network framework. One is in identifying differences in edges. So this is a paper from my group in 2015 where we compared networks inferred in two different subtypes of ovarian cancer, grabbed the edges that were most different and said, aha, here's what we think is driving these differences in these subtypes. Another is looking at differences in the nodes, such as differences in targeting. We look at something like a centrality score, a betweenness, a degree centrality, etc. And you basically say, okay, so the genes are targeted differently in these different networks. Maybe they're corresponding to different pathways. Maybe those pathways are important for disease. And I wanted to make the point that these two examples on the left are pairwise analyses, but they don't have to be. This is a paper from 2021 from my group where we actually did 38 different networks, and we did 38 pairwise comparisons to say what's different in each particular tissue and said what are the pathways in each. So the other way we often think about network comparisons is not just two groups. I talked about how we have 30 different tissues, and Mona said very nicely, what about different individuals, right? We like to think about, you have subtype A or subtype B, but sometimes different people have different ages, different BMIs, different sex, different covariates, maybe they're smoking or not smoking, etc. So unfortunately, most of the algorithms we have to generate networks, at least with omics data, things like transcriptomics data especially, generate one network. And that actually represents information from all the different input samples. And if you have two really well-defined phenotypes, maybe you can take omics data from one phenotype and omics data from another and compare them and get something. But in reality, everyone's just a little bit different, right? Even going back to that example of Susan and Jane, they're just a little bit different. And so more recent approaches have tried to basically take some of the outputs of these algorithms and try to generate networks for individual input samples. And I wanted to highlight two here. There are some more recent ones, but they're mostly on the bioRxiv these days, so I didn't want to throw them out there yet. These are two methods that have been published for estimating sample-specific networks. they have the same basic underlying approach. Let's say you have an algorithm to infer networks using all samples in a data set. You can apply that algorithm to all samples, and all samples minus 1, and then use mathematical trickery. It's not really trickery, they both have very well-established derivations, to try to reverse engineer whatever that sample-specific network is. And why is this important? Well, this is important because it lets us get into some of the nitty-gritties of heterogeneity. So I was going to throw out one example. I'll go through it quickly for time. This is an example driven by someone I work with, Camilo Lopez Ramos, on sexual dimorphism, which she was comparing males and females. Of course, this is important to understand the difference between men and women for health and disease, but often the women recruited in a study might be younger, they might weigh less, right? We know those differences are really men versus women or having to do with fat content and BMI, right? So what she did is she basically did a differential expression between all men and women where she could actually model all sorts of covariates, things that were important. And this is standard bioinformatics analysis, has nothing to do with networks. But in the context of networks, we only had one network for men and women most of the time. But she was able to actually apply the Linus approach and got an individual sample network. And then what she could do is she could run the same basic statistical equation to find edges that are different between men and women. And she could compare that to say, okay, here's the differential targeting, the differential edge weight as a t statistic against the differential expression, t statistic, and actually find correlations that way. And this is one example where there's still two groups, right? But it doesn't have to be men and women. It could be disease healthy. It could be a continuous variable, like something like pack years of smoking or something like that. And so that's kind of, I think, where networks are going. And I'm going to actually turn it over to Holly next. And as she walks up, I'll say that I think one reason why this is a nice transition is Holly's going to talk about dynamical networks. And if you have sample-specific networks, you can start thinking about how those individual networks actually change over time in a dynamic sense as well. So I'll turn it over to you. Did I do it the wrong way? Sorry. Yeah. No.
No, you need the other remote.
Use the other remote.
Slide. They're completely other remote, not that remote. On the table. There's another one.
This
That was
one?
the other one slide, yeah.
Oh, okay. Thank you. Okay, cool. So as Kimberly has introduced, and we see lots of examples in today's talk workshop, is like we're talking about networks, right? And when we try to analyze the difference between two networks, the object of our analysis is the moleculars representing different conditions, right? And when we see these different conditions have lots of connections, because we see, even when we're focused on difference, there are lots of nodes and links shared between this differential network. So when the different condition can be assembled along some orders, for example, time course, then we're interested, the new question is how this transition from one stable condition translated to another stable conditions, or what's driving this kind of transitions. One particular example often cited in biological science is this Wallington landscape in the development biologists, right? I want to emphasize here is when we say stable condition, it's kind of a group of a system. For example, the system means cell, single cell, or individual, could be both of the case. And with stable state, that means the system is stable, is resistant to small stimulus, right? You can see it still keeps what it is. Okay, that means stable. And but we all know more and more evidence shows in the biological field there also exist lots of transition states. This transition state currently popular talk in the biological field are two types. I'm saying it's reviewed last year. It's talking about pitchfork transition. That means the earlier state can be translated to multiple options, and this subsequent multiple options has a little competition. And another mostly often cited term, we call it set node transition. They have a beautiful example in clinical biology. It's like this, Is this like in the middle? This pre-disease stage, right? It's in translation of bioinformatics, we call it early warning signature. We want to make a diagnosis when the disease can be obviously diagnosed. We know from the genome omics data, we can find some early warning signature. Another example very well cited fitting this scenario is in development biology, when the progenitor cell committed to some fully differentiated cell. By commitment, that means this transition cannot reverse, okay? So, and computational work to study this transition, currently, in my knowledge, there are two types of methods. One is what we call the energy-based approach. That's based on the observation. When the transition happens, the system are unstable, or at least a group of molecular unstable, you can observe the increased local energy, or we call it in different words, you name it, perturbation, something like that. Another method, which my group currently is interested in, is based on this tipping point theory. Like I exampled before, the tipping point theory means this transition is not only irreversible, but also very quickly, abruptly. And we know when this transition happens, they have two features. First, a group of molecules gain new links among them. Second, the overall system-to-system similarity disappeared. That's two features. Based on the two features, we have these tools developed. And okay, I want to emphasize a little bit here. You can see from the, how can I do this? Okay, fine. Let me see here. You can see this nodes, we have the shared nodes. But in the earlier stable conditions, right, the links between these, what changes the links between these nodes. That's very interesting to study, and I know my biological colleagues are interested in, among this one, who, for example, in my example, in development, methadone development, this ETV2 only is a transcript factor, only abruptly depressed in this earlier progenitor states, then it shut down. It plays a kind of role to module the transition states. Its performance and its link towards its potential targets decide the future. What's the potential next stable states? Compare these two types of method, right? And we can think about a different angle. For one review thinks about it, we think, but I believe that coexistence in biological system. The transition could happen in a very, very slow, continuous model, or sometimes, like my example, this ETV2 module, the regulation subnetwork, happened suddenly, irreversible. So this energy-based model can be applied to both cases, but the tipping point-based model only, study this sudden, irreversible translation. However, in our recently studied 6 single-cell level the time course data developing development biology, we see this. We apply these two methods to the same data sets. This result converged on the same prediction of when the transition happened. What makes them different is what driving this transition. The predicted potential driver role of transcript factor is a little different because our theory or our focus are different. Whatever we describe, the feature is different. Finally, yeah, it's too hard for me to ending this, but for the future direction, right? I believe everyone here have your own sense of what will go. Here's only my own little opinion. Is kind of we all trying to solve problem when the new technology helps and new data come to us, and spatial and time course, temper data is increasing, and we're trying to fit our model for it. And you can read all these things and infer causal network. We believe, and repeat today's morning's talk, is another very important question to go. And I'm happy to, as everybody joins discussion, go to a real future direction conclusion here, rather than my simply list. Yeah, thank you. Why don't you stay up there? My tool. Being me coach talk. All right, so those are our slides in our presentation. I'll leave these last questions up in case people want to spur, and I think I should invite my fellow group members up here for the discussion.
So we started this session 5 minutes late,
Yep.
so we're going to go with discussion until 3:05.
Sounds good, thank you for comping us that extra 5 minutes. Are there any questions from the audience or comments? Yeah.
I have a sort of clarifying question to Lenore, if she's there. Namely for the sequence analysis.
Yeah, Lenore, are you there? Can you hear us?
Okay. Lenore, I have a quick question for about using sequences for the predictions, especially going between the species. I just wanted to clarify, whether any sort of multiple sequence alignment was used or just using the individual sequences, and what do you think interplay between using just single sequences versus multiple alignment would be for the problem?
Oh, that's a great question. So the current versions of both Descript and Topsy-Turvy only use single sequences. They do not need a multiple sequence alignment. It's clear to me that if we did incorporate a multiple sequence alignment, we would do better. But we were really, really laser focused on things that were fast, and so we could run them genome-wide. But we're definitely going to look at multiple sequence alignments in the future as well. I will say, though, that saying that it doesn't use multiple sequence alignment at all is a little bit disingenuous, because it is starting with a pre-trained protein language model. And those individual sequence models are certainly leveraging everything we know about protein sequence space, including multiple sequence alignments in some way, but it's a very second order effect, right? Your input is just the single sequences.
Thank you. Why don't you do first and I'll do? Yep. Yeah.
So, there's, now that we've had this hypergraph, we started off with hypergraphs and then hyperpathways. And I know that for hypergraphs there is centrality and in between this and so forth. I mean, how much of this can be applied? I mean, have we thought about going there? I mean, I feel like beyond that, my brain gets a little confused because I have trouble interpreting hypergraphs. But are these algorithms kind of the combination of network structure plus node weights kind of usable in a hypergraph context?
I can take one crack at this, and then we should ask Anna and Morali for the hypergraphs perspective. So we have one paper on hypergraphs where we actually started with this question, where we said, let's try to do something like network propagation on a hypergraph. And we started with the simplest form of the random walk on a hypergraph, which was your random walk's really on the edges. It's an undirected hypergraph, sorry. Your random walk's on the vertices. So you start at vertex, and then you choose a random hyper edge, and then among the hyper edge, you choose a random vertex in that hyper edge, and that's your random walk. Very natural thing. And there's a weighted version of this as well. You can choose your weights according to the weights of the edge. And what we discovered is that even though that random walk has been used in the literature for hypergraph prioritization and machine learning, was that random walk actually was equivalent to a random walk on a regular graph, a simple graph, a non-hypergraph. And so it wasn't actually encoding higher order information. It was actually equivalent to just different weightings of this pairwise graph. So we ended up writing a machine learning paper, it was at ICML in 2019, on this issue, saying hey, some of these hypergraph representation learning papers that use this particular version of the random walk are not actually modeling higher order interactions at all. So one has to be careful. And I think if you do directed hypergraphs and you do the random walk, what we showed is that if you do the random walk in a different way, where you actually sort of have weights on the vertices as well, then that couldn't be modeled by a random walk in a graph. But it's a subtle thing when you get into these propagation approaches and how you're supposed to sort of propagate information on the hypergraph, if that's what one wants to do sort of the equivalent prioritization.
Yeah, I could add one point. So if you only do the propagation on the regular graph and hypergraph mathematically, yeah, I can equal.
Yeah. In certain contexts, you have to...
Only for propagation, not for others.
Yeah.
That's a technical point.
Yeah.
But isn't that confusing in the sense that there are theoretical results that say that you cannot get the same cut between hypergraphs and graphs?
Yeah.
And these propagation techniques sort of depend on the cut. So how do we reconcile now these things?
Yeah, so it all comes down to the specific version of the random walk you do and how that's modeled by cuts. And there's a whole literature on this. There's a literature in, what do you call non-hypergraphs these days? Just regular old graphs? What do we call them? When they're simple graphs? I mean, there's...
No, not simple.
But not simple, because that means something else. So remember, non-hypergraphs, just pairwise graphs, the old... Yeah. So there are those parallels, and there's a literature on that in binary graphs, pairwise graphs, and where things were... In hypergraphs, the literature is more complicated, and the correspondence between the random walk and the cut is a little more subtle, and depending on, there's all these different ways to do the random walk on the hypergraph, and that was sort of some of the challenge. So the theory is much more complicated.
Why don't you, Gianna, and all?
So I got a question for Ben and possibly Sarah and Wade as well. So once one gets scores on nodes by combining the data and then identifies a subnetwork, typically how, to my understanding, the methods are evaluated is enrichment of nodes in the subnetwork in something. But say that the underlying network between the given set of nodes is a quick. You can identify all the edges or any subsets of them that connect all the nodes. How do we evaluate subnetwork rather than just set of nodes in the subnetwork?
I think most people evaluate the nodes in the subnetwork. Yeah. And hardly, I mean, in a sense, they're sort of equivalent because the nodes induce the subnetwork they induce.
If you think induced subnetwork is biologically important, but
yeah.
Say that the actual induced subnetwork is a clique, you can take any star or a cycle or anything and it's different subnetwork, which shouldn't be all equivalent to each other. So. Yeah, well, so I think what maybe you're pointing out is another version of like how identifying the subnetwork, altered subnetwork, is different from identifying the nodes in that subnetwork. Because sometimes what you really want to think about is identifying the nodes and the edges, right? Because it's not induced. To my knowledge, at least, and you guys should correct me, I don't know of anyone that in the kind of propagation and sort of these type of communities actually makes that distinction. Because the algorithms themselves mostly are not making that distinction.
I think
so one possibility might be to take edge annotations. For example, when you know that an edge is a part of the pathway and use those for adjustment rather than doing nothing.
Yeah, I just to follow up on Ben's thing, I think it depends if you think about it as subgraph identification or if you're thinking about identifying signaling pathways or things like that. And so in those fields, people do think about whether you have the right edge or not, right? Because it's representing some kind of causal relationship.
And just quickly to add to this, often you have weights on the edges, and then those weights heavily influence propagation methods.
Yeah, sure they do. I guess what I'm the point I was trying to make was that most of these methods, when they propagate, it's really just vertex. They're just changing the vertex scores, right?
So they can output non-induced subgraphs, but they evaluate just the nodes, I think.
Most people evaluate the nodes. The question is, could you pull out a subset of the edges? Like once you've sort of decided that you want this set of nodes based on your ranking or based on whatever you do, like some sort of post-processing ad hoc thing, I suppose you could, in a way, pick a subset of the edges based on these vertex scores, based on the original weights of the edges. I should clarify, probably someone does do this because the literature is vast,
I think it
but
depends
it
on
doesn't cover it.
application, right? So if you're trying to determine causality in these networks, like how does a signal propagate through the networks, and you care a lot about the edges. If you're trying to identify, sets of proteins associated with some process, then you don't care so much about the edges.
Yes.
And so I think depending on the kind of application, people have done different things.
So how about the different neighborhood?
Sorry, I don't know who's monitoring.
Okay, so my question is how about the PageRank neighborhood? Like you do PageRank random move that is starting from the seed node to give you a subgraph around that seed node. So it's like a random move without a restart. It always starts from the seed node. So eventually not only you get the ranking list, but that actually gives you a subgraph around that seed node.
That's the random walk.
Based on what? It's just.
So it's like an approximate PageRank algorithm
Yeah.
that gives you the sublinear compact because you only start from the seed node. So what it does eventually it gives you is a subgraph around that seed node.
Oh, how does it give you a subgraph? It'll just, like any version of network propagation, will just give you some new scores on the vertices.
Yes.
And then you have to interpret
So
what does
how
that
the
mean.
iteration gives you a cut, right? So based on the propagation score, it gives you a cut. That seems to start from certain seed node. So that cut itself will give you some subgraph. Yeah, I can give you the.
I mean, yeah, sure, there was, I think there was a
Maybe
comment.
like tie a couple threads and then lead to a question I had for Ben. So, Mona mentioned context is important. So when we think about signaling pathways, we're very interested in Tiana's question of did we select a reasonable set of edges? So these max flow things and signer trees, that whole family of methods, it really does want to select edges sometimes. And we've struggled with trying to evaluate that in new systems. Maybe we don't have a tag pathway or some gold standard to evaluate against. And we've started to poke around looking at like what are the types of edge topologies that are completely unreasonable, because we saw this morning how we can characterize topologies maybe with like these graph-like count vectors, the subgraph counting things we saw. And sometimes I can say like, if I have a star graph where I have a clique, some of those things are very unlikely to be what I expect my signaling pathway topology to be. I think there's probably ways to rule out really bad things. I'm interested in trying to figure out better and better, like leading to what Ben was talking about, what is the family of subgraphs that's plausible in biology or in signaling? And I guess the thing I struggle with is, it's probably something that's really difficult to characterize. And you were listing a few options like connected graphs. Those are fine, we know how to work with them, but I would bet there's some awkwardly shaped family that's probably realistic, and I just have no idea how to work toward defining that.
Yeah, and to be fair, I mean, the ones I listed were just ones that people have used and could be written down. And I completely agree that probably the correct one is some weird thing, but then, then there's some strange circularity, right? Because if you can never design an algorithm to, I mean, maybe you can. It's the algorithm has some topological bias that is representative of biology, but you just don't know what that bias is. You can't sort of write it down. Yeah.
I can only half hear the questions, but I would add that in many domains, we have so many missing edges that it's a little bit dangerous to be making topological conclusions. Or if you're going to make strict topological conclusions, you should always remember you're seeing a sample of the network that is not complete in this case.
I have a question about comparing networks, and maybe this gets into differential network analysis and also maybe a little bit about network alignment. So when we introduced the problem of network alignment across species, the goal was to find conserved and divergent networks. I was curious, like if you have networks across individuals or conditions or whatever, how do we actually go and compare easily? So we could compare the networks and say, okay, here are the set of edges that are different or similar. But how do we get to like higher order types of differences? Can we do that? Like, can we take that? One more thought.
Yeah, I can try to take a crack at it. You touch a number of things. One, comparing species, networks from different species is going to be different than comparing networks, say, within the same species, because different species will have different sets of genes or different sets of molecules. When you're in the same species, you know, the issue of alignment is a little easier in that you have at least the same set of nodes, and you can find the differences in the edges. But like you said, once you get those differences in the edges, kind of what do you do with them? Especially in inferred networks where they're very noisy and you don't have a lot of confidence in a lot of the edges. So what I often see people do is they take those different edges and they often reduce them back to the nodes somehow, right? So you find a set of edges and you say, okay, here's differential, you know, the set of edges are all tied around this hub protein in this one network and not in this network, and so maybe it's important in the disease context versus not. There are some algorithms I know of to look at differential community structure, including on inferred networks, to find, say, differential modules in a very robust way. And then, again, how do you evaluate that? You say, here's a community that's in one network but not the other, or shared between the two. You tend to still reduce those to pathways and do enrichment. Does that kind of answer your question?
And I was also wondering, you know, you could think about, like, if you want to take a graph, different graphs, and encode them, and then try to, you know, compare them, and graph neural networks down to recipes and stuff. I'm looking for, like, you know, ways to go beyond just edgewise comparison. And, you know, can you say something about the interesting substructure without, I don't know.
Yeah.
Is it possible, like, you know, looking at the dynamics of networks and within the same species, but try to find, like, higher order differences?
Yeah, so it's a great point. So what we see in ML literature in the last few years, there are papers published that focus on embedding subgraphs, but they are more or less focused on on a subtle problem, which is you start with a graph and a set of subgraphs that you think are interesting, that you found somewhere, and then you can embed those subgraphs. So that is useful to the extent that once you embed them, you get this condensed data summary, so they become easily comparable to each other, because then you can compare those vectors. But it's not solving the problem of, can you take a graph and can you find those interesting subgraphs yourself? That would still require you to, like, either exhaustively enumerate subgraphs or use random walks or some other clever techniques to find interesting subgraphs before you can embed them. So I can think of a handful of papers that are trying to find, or essentially come with a differentiable counterpart of what an interesting subgraph would look like. Essentially come up with an approximation, an objective function that approximates a subgraph that is overrepresented in the graph, and then declare those subgraphs to be interested. But in that regard, it's always a problem to define what is an interesting subgraph. And we go back to lots of questions that were mentioned today. So it's really not a magic bullet to solve those things, right? It's primarily about, well, if you understand the problem well, perhaps it's really hard, you can come up with some differentiable approximations to this problem, but fundamentally it's still about, like, deciding what interesting subgraphs are. It's a bit more challenging than embedding subgraphs themselves. That's quite advanced, because now we can, essentially what a neural net would allow you to do is to find good aggregation functions. Ultimately, it's still about, like, often learning these embeddings at the node level, but then integrating them in a clever way, not by simply taking an average of node embeddings or edge embeddings, but perhaps upweight, downweight some of them based on other type of data or based on topological measures.
So in terms of look, there's a lot of stuff in trying to find interesting subgraphs in terms of enriched subgraphs, mostly based on enumeration. And so whether that's, like, just topological based patterns, or, you know, as I mentioned, combinations of topology plus annotations. And I think actually the hard part there is setting up your background null distribution about what's interesting. So, like, I claim that if you're looking for higher order structures, that you should control for the frequencies of the lower order structures. And so that's actually really hard to do. So how do you know, look at a subgraph and say, you know, I want to make sure that all the triplet combinations that I saw, that distribution is maintained in the graph. And so as you move to, you know, bigger subgraphs, it's both like an enumeration issue, plus also an issue of how do you set up your, you know, your background model. So I think it's actually pretty tricky.
Could I make one quick comment, like a terminology thing that would be great to sort of resolve in this meeting? There's higher order structure. And so I've always confused whether higher order structure is supposed to mean a hyperedge, meaning it's an interaction that's more than two things, or a clique of 3 nodes could be higher order structure.
Both.
Some comments, I want to causing all the members' attention is we often talking about overrepresentation when we infer some knowledge from the networks, right? And that's a little biased based on the data available to us. Current measurements is measured mostly the stable things, right? And also the overrepresentation means the critical signature pathway is stable from earlier state to later state. They're shared. However, biologically, they are more interested in who is a small number of them play the role to decide the future, or the small one you can translate the people's response to some drug, something like that. They're not interested in the overall, the genome background, who is shared. They're interested in this typical, very specific one, which might not have the feature of overrepresentation in the data we currently observe. But how to solve it? I hope I hope everybody here expects in computational field can help me. Yeah.
So let's do one more question, and then we're gonna start the coffee break.
Yeah, just really quickly, Mona, Holly, and the speakers talked about virtual networks and talked about different from people, person to person, and different conditions. I just want to add one thing is it's touched upon, but make it even more, I guess, obvious. Is just, but when we look at two people, they don't have one. Each person don't have one network because different cell types will have, right, all the different networks. So I just think that something should be. I just want to mention that because I think that's very interesting. We just had a paper recently looking at cancer mutations. By looking at specific cancer type and construct a network in that cancer type that have a better performance than just taking the overall and analyze all of that. So, I just want to.
Yeah, we just need so many samples that are individual specific and single cell specific, and Well, thanks, Kimberly and the team.